So today we're looking at how an insertion or deletion of a single base pair in an mRNA sequence can lead to large variation in the resulting amino acid series. Having a universal genetic code codon chart while working these problems is necessary. All of the codon options come directly from this chart. I've included a link to a Khan Academy page that has information about genetic code as well as the codon chart in the description. Determining the mRNA code of the first two amino acids is as simple as reading the chart because both starch/methionine and tryptophan have only one codon possibility. Next, we see that the normal protein has phenylalanine as the next amino acid, while isoleucine comes next in the mutant protein. There are two codons that code for phenylalanine and three codons that code for isoleucine. Comparing them side by side, we see that all of the isoleucine codons begin with an A, while none of the phenylalanine or tryptophan codons contain an A. Therefore, an insertion of an A base adenine must have occurred. So we can add an A as the next base in the mutant sequence. In order to determine which of the possible codons are part of our sequences, we need to consider the next amino acids. We know that the first two codons of phenylalanine have to be UU, which means that the codon for isoleucine must be AUU. Now, in order to determine the third codon in the normal protein, we look at the next amino acid. We see that both possibilities for histidine start with C, so UCC is the correct codon. We already have a C in place for histidine. We can see that all of the options for threonine begin with AC, which reveals that the mutant codon for histidine is CAC. In order to figure out the third base in threonine, we need to look at the next step in the mutant sequence, which is stop. All of the codons for stop begin with a U, which tells us that ACU is the correct codon for threonine. The next amino acid in the normal series is lysine. Both of the codon possibilities for lysine begin with AA, which gives us UAA as the stop sequence that determines the mutant protein. The third base of the lysine codon and the codon for glycine cannot be determined from the mutant protein as it has terminated, so we will just add any of the possible correct codons. And there you have it. We have worked through the problem using the normal and mutant amino acid sequence in order to determine the mRNA sequences of both proteins.